Welcome to Mr. Chalk's Revision Tips. In this video, we will look at photosynthesis, the leaf structure, and testing leaves for starch. So, if we remember from previous videos that photosynthesis is a process where carbon dioxide is reacted with water to make glucose and oxygen, and that leaves have some general adaptations. So they have a large surface area, there's lots of them to increase the surface area, they're thin so gases don't have far to diffuse, they contain chlorophyll, they have good networks of veins to supply water and to take the sugars away from them, and they have stomata which allow gases to diffuse in and out. So, when we're looking at practical activities in science, it's important to remember some key words and such forth that we need to use. So a control variable is any sort of variable that does not change during an experiment. A dependent variable is the variable that responds to the independent variable. So the dependent variable is the one being measured. A hypothesis is a prediction of whether the independent variable will have an effect on the dependent variable. The independent variable is the variable that is manipulated or changed by the person who's doing the experiment. An anomaly is an observation that differs from what we would have expected to have happened. And a correlation is the relationship between two variables. An error is a measure of the difference between the measured or calculated value and the true actual value. And precision and accuracy are often used interchangeably, but in science they must have very different meanings. So measurements that are close to the known value are said to be accurate, whereas measurements that are close to each other are said to be precise. So when we go and we look at exam questions and such forth, there are lots of them that look at how we go and we test leaves for starch. So we've got to remember that the plant converts glucose into starch because it can be stored more easily. If the plant left the glucose as just glucose, it would be water soluble and would get washed away. So you can test to see if a leaf has been producing starch, and we can look at describing patterns and such forth in what we see. So if you remember, photosynthesis can be investigated to show the production of starch and the importance of chlorophyll. So when plants photosynthesize, they produce glucose, which, like I've just said, is then converted into starch. We can then test the leaves using iodine solution to see whether any starch is present. So this is because the iodine reacts with the starch and turns a black colour. So iodine, before it's done anything, is usually a yellowy brown colour. So when we're testing leaves for starch, these are the general steps that we think about using. So we start off by boiling the leaf for 30 seconds in boiling water. So this stops any chemical reactions taking place. So it's stopping any of the starch from being broken down. We then go and leave it in boiling ethanol for a few minutes. So this boiling ethanol will go and it will remove the chlorophyll. We then wash the excess ethanol off of the leaf in some hot water and spread it onto a white tile. And finally, we add iodine solution onto the leaf using a dropping pipette, so the parts of the leaf which turn black will contain starch, so we can see that starch is present. So variegated leaves have green parts, so where the cells contain chlorophyll, and white parts where there is no chlorophyll. So only the parts where the green become blue-black with the iodine solution showing, or it shows where the chlorophyll is present, because that is where the photosynthesis will have taken place. So the plant can be de-starched by leaving it in the dark for a few hours. So parts of its leaves can be covered with dark paper, and the plant is left in, well, the other part of the plant is left in the light for a few hours. You can then uncover it and test the leaves separately to see if any starch is present in them. And we should see that the plant that has been in the dark will have a much lower concentration or much fewer areas where starch is present.
Thanks for watching.